Microsoft, working on proteomics, genomics, and public health.
Yeah.
Is that correct? Yes. Right. So thanks, Meghna, for making your visit. Look forward to hearing you.
Thanks a lot for having me here, Ganesh. It gives me a lot of pleasure, and also I'm very honored to be here. I never thought I would be giving a talk at IIT when I was here as a student. So I'm in the AI for Good lab at Microsoft, and like Ganesh said, I work on a number of different areas, but my favorite topics are uh, at the right end of the spectrum. So I work at some macro level topics like public health, health outcomes and equity, also on statistical genetics. But I also work at the uh, micro scale, at the molecular scale, where I've done work on modeling DNA and RNA sequences, on modeling protein sequences and protein structures. And today I'm going to be talking mostly about this part of the work that I've done. And so, at a high level, I'll talk about modeling protein structures using graph neural networks, where I'll introduce one open problem. And I'll also talk about some recently published work on using DNA sequencing data from next generation sequencing technologies. Before I begin, I want to get a rough idea about, how familiar are people with proteins or with DNA, or would you like me to start from the cell? Maybe a quick show of hands for how many people know about proteins. Oh, okay, so that's a lot of people. So I don't need to go into details of what a protein is and stuff. Okay, so I'll start with learning, this model for learning transcription factor profiles using variational autoencoders. Now, as we all know, our body has a large number of cells of very different types. They all start off as stem cells in the bone marrow, and then they differentiate into different types of cells, and they migrate to different parts of the body. So, but then at the end of the day, every cell still has the same DNA. But then, as you know, blood cells look very different from neurons. They have very different functions. And the reason this is true is that proteins called transcription factors, they control the fate of a cell. So when the cell differentiates from a stem cell into a neuron or into a blood cell or into a muscle cell, these proteins called transcription factors, they turn some genes on and off. And the genes that are turned on and off determines what the cell eventually becomes. So, and these on and off events, they happen at locations where the DNA is open. So if you look at this cartoon representation of this process on the right side, here's a cell, here's a chromosome, and if we unwind it a bit and look at it closely, is the double helical DNA structure, and here's a protein molecular transcription factor. The molecule, this transcription factor probes the DNA at different places. It has a favorite pattern that it likes, and once it finds that pattern, it'll go and bind to that location. So here it probes here, it doesn't find it, then it comes here, it finds the sequence on the DNA, TAATA, that it likes, and it binds here. So as a result of this binding here on the DNA, some gene either maybe here upstream or some gene maybe downstream will be turned on or off, and that influences the cell's behavior. So summary of this slide, transcription factors are DNA binding proteins. They only bind to the DNA. They bind to specific patterns of DNA. So they do, like this transcription factor, if it finds say ACGT somewhere, it's not going to bind there. It only binds to T8ATTA, and they control the fate of the cell. And the key thing to know about transcription factors is that when they probe the DNA, if the DNA is closed in this looped form, where it's bound very tightly and coiled up, then the DNA sequence is not accessible for the transcription factor to go and bind to it. So the closed parts of the DNA, you won't see these proteins going and binding there. On the other hand, the open parts of the DNA on the right here, as you see, those parts of the DNA, they are unwound completely. The DNA sequence is available. It's easily accessible. There is no competition. And the transcription factors here, so I've shown them two transcription factors, one in pink and one in green. They are both going and, because this region of DNA is open, they go and bind there. So now the thing to understand is that, I just told you that what determines cell-fate is which genes are turned on and which genes are turned off. And now we are taking one step back. We know that which genes are turned on and which are turned off is determined by which regions are open and which regions are closed. So based on what is open and what is closed, the genes will be turned on and off. So now, if we were to develop an assay that tells us which regions are open and which regions are closed, we'll be able to get a good insight into what is it that is different between different cell types, what is it that results in cancer, for instance. And there are a number of assays that, laboratory-based methods that let you find out which regions of the DNA are open and which are closed. So the way these assays work is you break the nucleus, you extract the DNA is fragmented, and then you use this sequencing technology to find out the open and closed regions. So you sequence, the way the assays are designed, you only end up sequencing parts that are open. You're not able to sequence parts that are closed because the DNA is just not open and available there. So here's at a high level, the types of assays. There's many different types. I won't go into details here. I can come back to this if people are interested. But now coming back to the question. So what we want to understand is which regions are open, which regions are closed, and what is bound there. Because the transcription factors are the ones that are keeping the DNA open here, right? So the reason the sequence DNA is open here is that there are transcription factors bound there. So this is a very broad open problem in the epigenomics research, in epigenomics research, that we want to know where are these transcription factors bound, because that's what controls cell fate. And the way you can do this from DNA, accessibility data, so that is where DNA is open and where DNA is closed, you get this type of a signal from the experiment, from the laboratory experiment, where this is the strand of the DNA, ACGT, and so on. This is chromosome 19. So I'm showing this particular location of chromosome 19 zoomed in. And this is the DNA sequence at that location. And the signal I'm getting from the assay is just that this is open and this is all closed. So wherever there is like this peak, wherever things are peaky, those are all open regions, and these are all closed regions. So now, given such a readout from the experiment, what we want to do is infer what is being bound at each of these open locations. So, so if you remember the slide that I showed here, right? So, these two transcription factors were sitting here, and we want to know what is being bound. So that's what we want to do. So here I've shown three different proteins, three different transcription factors, CTCF, BATF, and PU.1. And in this cartoon depiction, what we want to do is, given this sequence, find which one is bound where and what pattern does that protein like. So there are two problems here.
That's not one. What's the length?
So it depends on the assay being used. Typically, the length is something around, it depends on the size of the DNA fragments that are generated by your protocol. So it can go anywhere from around 50 base pairs to a few thousand base pairs. So 50 to a few thousand is the length of the DNA sequence. And when you process the data further, the size is usually a few hundred. So it can go from like 50 or so to maybe around 500. And typically most methods use around 500 or 1,000 length sequences to build models. Yeah, so feel free to interrupt me at any time with questions. So there are, what the assays give you are two things. One is the location, where this is. So this is chromosome 19, offset 36 to, 200,000 something, and that this is open, and it tells us what the sequence is, where it's open. So what we get from the assay is GC, GC, AT, GG. This is the region that was found to be open. What location it is at. And now what we want to do is find what may be bound there. So now why is this interesting? Like, why do we care about where transcription factors bind and all that? So the reason is that this will enable real-time epigenomics. This is a pipeline that's often used in cancer immunotherapy, for instance, where you get some blood from a patient, you do these assays, for instance, the assay called ATAC-seq, which gives you these sequences, which are basically open regions of the DNA. So each DNA sequence of around length, 50 to 500, like I said before, that is the open region for that cell type. So for blood cells, this is what we know. So this is the
region means like throughout this length it will be open.
Yeah, throughout this length it will be open, exactly. And after that it won't be open. So once you have data like this, you can basically look for, okay, so this region is open in this patient, but normally this region is not open in most patients. So maybe that's why this, they have a certain type of cancer. For instance, here, like this is the gene NFAT, and a particular location on chromosome 4 here, this is open in this cancer patient, but maybe in normal people, it's not open. So then that lets you decide what immunotherapy drugs to use, because you want to use drugs that will go and close this location. Okay, so moving on. So the summary of the work that I'm going to show here is that variational autoencoders based on Dirichlet distributions, they can deconvolve this type of data. So given such just like these open regions of DNA, like a bunch of sequences, and you know that they are open, you can use variational autoencoders to get what is bound where. So so these are two proteins, ETS1 and another protein here, and this is the input DNA sequence to the model. What the model can tell you is where what was bound. So there are a number of different approaches that have been tried for this problem, going from convolutional neural networks to transformers. As you can imagine, you can take all the DNA sequences, you can think of it as a one-dimensional data, and you can have convolutional filters that, like move like a sliding window, decide a certain stride, have a bunch of layers, and then predict that this is open or closed. Or maybe you have data where you know that this region is open and that a certain protein is bound. So then it would be a multi-class classification problem where you have the DNA sequence as input and the output is the protein, the type of protein that was bound. So there are a number of different models. Yeah.
So I think when you train these VAEs, you use a default reconstruction laws, and when you talk
Yeah.
about transformers,
Yeah.
with the mask models, for example.
Yeah, mask models. Yeah.
There's a lot of self-supervision.
Yeah, a lot of self-supervision. So here, the VAEs that we use here, they are unsupervised, so we don't use any label data. But yeah, typically it's self-supervision. So
Data is just that gene sequence.
the DNA sequence. So the data is basically just the DNA sequence and a number of those DNA sequences. So is there a marker here?
Yeah, just beginning.
Yes. So the data will be basically some location, so like chromosome 19, and then some, and so on. So this will be, like I said, it would range from around 50 to 500. Typically, some methods that want to use context. So even if the region is not open, they might use bigger windows. So the data is all of this kind, like chromosome 19, then a big DNA sequence. Chromosome 5, offset. So say offset is X1, which is just some number. So let me just write it like, say offset 52. Offset is 555. Then sequence is GGCC.
What does this offset mean?
It means in chromosome 5, the location where that was open, the location where the open region starts, and then the sequence at that location. So there are basically 3 bits in the information. One is the chromosome number, then the offset within that chromosome where the region is open, and then the sequence at that location where it is open. So this is the information you have. And some methods use positions as input to the models. So you can think of the way positions are embedded in like transformer NLP based models. Some people, some previous work has used these positions to, you can apply like a sinusoidal transformation to this and use that as well as an input to the to whatever encoder you have designed. But in this work, we only use this sequence. And
this chromosome number, let's say that is chromosome 19.
Yeah.
That means it has a complete description and that is unique.
Yeah.
Is it unique for per person or is it unique for everyone?
So so across people, the DNA is very similar. So 99% of all of our DNA is the same. But there are some key regions that are not conserved. So usually the regions that are not conserved may or may not be sequenced. So it depends on what sequencing depth you are using and so on.
So when they are saying somebody says chromosome 19, so that chromosome 19 description is taken from a textbook. It's not the.
Yeah, it's taken from the reference. So basically, every person's, the way the sequencing technology works is each person's sample is sequenced and you align it to the reference genome. That's like the human reference gold standard. You align it to that, and that tells you for you what's there at chromosome 19 position 52. So this is likely to differ from person to person.
This number 52, the offset will depends on preparation.
Yeah, no, the offset would be dependent on what is open. So yeah, it depends on the patient in that way. That maybe this region is not open in my cells, but it's open in somebody else's cells. Yeah, go ahead.
So suppose if we get a sequence and find it out that these regions are open and this. Now, how are you finding out that what are the proteins are bound to that open regions?
Yeah, so there are before deep learning was used, what the methods that people were using were called motif matching methods. So where people have studied certain protein families for a long time, and so for instance, there's a protein family called homeodomain, and homeodomain proteins, transcription factors from the homeodomain family, they all typically like to bind to the region of the DNA that goes like TAA. This is the sequence that typically these proteins like to bind. So what people would do is you can build like a this is called like a motif. This is like a template. You can think of this as a template, and you just take this template, you match it everywhere on wherever things are open. If say here at chromosome 5, position 555, the sequence was TAA, then you're just matching everywhere. Okay, no match here. Okay, there's a match here. So yeah, found something like that. So earlier the methods used to be like this, like just match. But the problem with these methods was that you don't know these sequences for all proteins, and secondly, there can be a lot of variability across cell types. For instance, in neurons, you may see that homeodomains like to bind something slightly different. They like to bind TTAA, so just slight variations.
Another thing is that they say, so you said that this is open and some proteins are found.
Mhm.
Now the question is important here. How does that region become open? This is this 19 and 50
Yeah.
position that is open in
Mhm.
the cancer patients, and
Mhm.
you're saying that maybe the wild type or the normal patient, normal person,
Mhm.
this is closed.
Yeah,
Now, some upstream thing must have happened which
Yeah.
makes that open.
Yeah, totally.
That can be
yeah.
something.
So that can also be, that can be one reason why it's open. It doesn't always have to be that the TF bound is the one that's influencing. So to answer more concretely, there are proteins called pioneer transcription factors. So they are ones that go and create these favorable conditions for the DNA to open. And then some protein, like some homeodomain may come and bind here. So there are another type of transcription factors called pioneer transcription factors. They are the ones that do what you're saying, that they open up parts of the DNA in many places. There's another transcription factor called CTCF, which is also responsible for, like creating a lot of this structure. Okay, so maybe in the interest of time, I'll quickly. Yeah, okay. So the challenges are important here. So so within a peak, so a peak is basically the open region of DNA. There can be several binding sites. So a binding site is basically like this sequence. For instance, chromosome 555, this particular location, this is the binding site, right? So there can be several binding sites within a single open region. There's a lot of heterogeneity. So like I said, this pattern can vary a lot across cell types and even for a single transcription factor. And there are many many transcription factors that like the same sequence. So for instance, here on the right, I'm showing examples of both. So FOXA2 is a transcription factor that likes, the sequence at the top in some cases and the sequence at the bottom in some cases. And then here on the right side of the slide, ALX3 and ALX4, these are two different proteins, but they like the same sequence, more or less, TAAT and so on. So like how can the model distinguish all this, right? So these are all challenges. And what we have is a Dirichlet based variational autoencoder. One very easy way to understand this is think of like topic models. Are people here familiar with topic models by any chance?
Very few would be, most have healthcare background.
Oh, okay, okay. So basically, what the basic idea is that you want to look for groups of similar patterns that occur together, and then that group of similar patterns is all associated with the same protein. That's the high level idea. So the input to the model is this open region of the DNA. So for instance, this region where it was open and the sequence here, and that one is represented as a vector where you are counting how many times each k-mer appeared. So each k-mer basically just means like the DNA sequence of a certain length. So here I'm using 8 mers. You're basically counting each 8-mer, how many times it appeared in this location. So that's the input to the model. So just k-mer count vector. And then the hypothesis is that each sequence has a distribution of binding sites over the different transition factors. So what that means is at every location, there is going to be some probability of finding transcription factor 1 and some probability of finding transcription factor 2 and some small probability of finding transcription factor 3, something like that. So that's the it's like a mixture model if you've heard of mixture models. So so in this model we have an encoder, and then there's the bottleneck layer, which is the which lets us project the DNA sequence into a latent space. And the assumption is that once you project the DNA sequence in the latent space, they will all cluster based on what proteins are bound to them. So if a DNA sequence, if DNA sequence 1 and DNA sequence 2 both have the same proteins that like to bind them, then they will be projected close by in the latent space. Whereas if they have very different proteins being bound to them, then they'll be projected far apart in the latent space. Does that make sense?
What is that?
So here the latent space is like 100-dimensional. It's like a 100-dimensional embedding. So it's basically, it's called latent space because it's like a hidden layer, and you learn the parameters for the hidden layer.
So it's the input variable length or it is also of a fixed length?
It's variable length. I mean, that's the reason we picked this representation of k-mer counts, that it can be any length.
The zeros, 001s, what are they representing? One monomer.
Yeah, so one indicates the number of times that this k-mer AAAATTG was found in this DNA sequence. So it's just the count of this particular k-mer. And what the vector is basically all possible k-mers will be in the vector, and the counts will just be how many times each one was found.
This is based on the absorption of certain TF protein or something.
What is based on?
Like how did we get it?
How did we get this k-mer count vector?
Yeah,
We just count here. So for instance, I go here and see, okay, so AT, this appeared once in this sequence. So I just go, you just go and count how many times things are coming.
Okay, so all the possible protein types in the vector down
Yeah.
is written, and top the numbers are like what are the counts of that particular combination.
Yeah, of that particular DNA sequence, k-mer, or of that. These are called k-mers. So it's an 8-mer because the size of this is
These are
8.
combination of all the.
All possible, yeah. Yeah, so for instance, the number of 8-mers of size, the number of DNA k-mers of size 8 would be 4 to the 8 because there are 4 different possibilities for each position. Yeah, sorry.
I have a trivial question. Is
Yeah.
a k-mer same from left to right and right to left? Or
That's a great question. So, so reverse, it's called, like reverse complement. So it's not that simple. So when you do it reverse, there can be palindromes, and then DNA sequences are also, they're called reverse complements. So basically, if you take the reverse of a sequence and complement the nucleotide, the sequence that you get, it's assumed to be the same as the original sequence. And so each k-mer is counted multiple times, contributes to multiple things. Yeah, so it's a bit tricky. I mean, I can answer it maybe after the talk if you want. So, but yeah, we do count backwards as well.
We fix the K or?
Yeah, for this work, we fix the K. We choose A because, we choose 8 because it leads to a manageable space and computationally tractable.
since you brought up the analogy with the topic models,
Yeah.
we also observe burstiness here, like in topic.
So,
Maybe explain what burstiness is. I guess, do you observe burstiness at all in the?
sorry, I don't quite remember. What is burstiness?
The burstiness of words, right? So
Yeah.
certain words which are very,
Oh, you mean like stop words?
not
No.
necessarily, I mean, there's a tendency for words to keep occurring,
Right.
right? They occur in bursts.
Right. So there is certainly a tendency for some types of sequences to appear together, which is why the method does work, right? So,
Okay. But there are specific models that handle burstiness patterns, so you haven't.
no, I haven't
handled
explored.
specific-
No,
Okay.
no. So this is like this is also a variational autoencoder. So it's a bit difficult to, get it to learn reasonable approximations. It's not like I'm doing Gibbs sampling. So the lot of the probabilistic models that have been developed for topic models, they were built for a much smaller scale of data. And so if you, and they have like MCMC based, often it's MCMC based inference and learning. So what happens is it doesn't scale well to this. So variational autoencoders are like kind of being used very recently. In the last 3-4 years, they have been used a lot for different. So I haven't yet like seen a lot of more complicated probabilistic models being used, just because it's difficult to make them work very reliably, given that this is all variational inference.
So these vector representation, it's for the model
Yeah,
to make the input as compatible to the model.
yeah, exactly. So yeah, there's the bottleneck layer, which is like you know 100 or 200 dimensions, and then there's the reconstruction, which is done by the probabilistic decoder. And the assumption is that the the decoder contains these distributions, K-mer distributions, that tell us the types of K-mers that are liked by different proteins. So the assumption is that the latent space, each component of the latent space corresponds to one protein's preference. Does that make sense? So, like say there's protein one, one's preferences are all in component one, protein two are all in component two, and so on. By preferences, what I mean is the protein, which ones are like less. That distribution is what's being learned by the decoder. That's the assumption. So yeah, so we make this assumption because typically. Sorry, what was the question again? Just is there a reason for
assumption?
okay. So the reason we assume this is that usually, if you look at the patterns, that these motifs that have been learned by previous, rule-based approaches, you see that typically there's like some variation in the sequence, but it's not the case that it's going to be, dramatically different. So most patterns that the protein will like to bind would fall within a few edit distance from this, like maybe 3 or 4 edit distance from this. Proteins that have very different patterns that they like. And in those cases, we find multiple components corresponding to a single protein. So there are some such exceptions. And there are also cases where the protein likes a very big sequence that it likes to bind to, like the motif is very long. So here the size of the template that the protein likes is 6, right? But it can be pretty long, so it can go up to 15 or 20 in some cases. But then because our latent space depends on this like K-mer size, which is 8, we can't capture everything in one component. So what happens is the beginning part of this 15 is in one component, and the later part of this 15, so 1 to 8 would be found in one component, 9 to 15 would be found in another component of the.
You are correct.
Okay.
Have you unmuted yourself?
Yeah, I have unmuted.
Alban, could you ask a question?
Yes, maybe you answered it already, Meghna, but
Yeah.
I was just wondering, typically in topic models,
Mhm.
you still have to figure out the semantics of the topic at the end,
Yeah,
right? After you
yeah,
do the model.
exactly.
So
That's a
similarly
great question.
here, you need to figure out what the transcription factor is for that particular family of sequences, right?
Yeah,
How
exactly.
to do that?
Yeah, so I'll
Yeah,
come to that procedure. So, so the inference is the objective and optimization is like this elbow evidence lower bound, and we use so a lot of the VAEs out there are Gaussian VAEs, and they use the reparameterization trick to propagate back propagate gradients of the stochastic bottleneck layer, but then it doesn't work for the Dirichlet distribution. So we have to use like this approximation, and we use some rejection sampling based, and we use some inverse CDF of gamma based approach. So let me move on to the question that Alpit just asked, which is, so we have these latent factors, and then we have these transcription factors. And like he said, can you hear me?
They can hear you.
Oh,
Your screen not.
Could you share your screen, please?
oh, screen
yeah.
is not
can't see the
sharing.
screen. Yeah.
Yes.
Okay.
But it's back to. Okay. It's still something visible. Just do a complete screen share. Just too small.
Yeah, I think if I do a complete screen share, somehow it's sharing a different
Oh, I see.
window. So let me try again.
Yeah, this is fine.
Okay. Yeah, so to Alpit's question, so we have these like latent factors which are like the topics, and we want to know what each one corresponds to. So we have an algorithm which is based on some hypothesis testing. And the way the algorithm works is we look at all the, we use a different source of data. So, so this data, the model that I showed you, the variation autoencoder, that was trained on DNA accessibility data. And we use another source of data, which is comes from a different experiment. And that experiment tells us what types of sequences are liked by which proteins. And so what we do is we do a hypothesis test where we look at where we take those sequences from the parallel source of data and we pass them through the model, and then we see which component likes which which sequence. Does that make sense? So I have a slide on this, but I don't have it in the deck. So we use a parallel
makes sense.
source of data where we have 20 base pair long sequences, where we know which sequences are liked by which protein.
Yeah,
So
I
we
was
have,
wondering how you could do that using the DNA data alone. So that's what I was curious.
yeah. So that's a good question. We use a parallel source of data, which is not like a ground truth, because that's not done in cells. That's done in synthetic DNA. So that's why it's not a reliable source of data to train a model, but it can be used to infer what the topics are, to match the topics to the proteins.
Thanks.
Okay, so moving on. So we analyze a lot of the results and we compare it to some prior approaches. But in the interest of time, I'll skip some of these final results, but I'm happy to share the slides with Ganesh, and if somebody wants to look, they can take a look. And then, of course, the paper is also published. It's in Genome Biology. Let me move to the second part of the talk. And so how much time do I have?
Maybe 10 minutes.
Okay, so I'll do a quick. Okay, so this is more standard structural biology sort of work here, where what we want to do is, given a protein and given its protein structure, we want to know if it might open up in some space, in some locations, and make some pockets available to us where a drug can go and bind. So I'll skip this slide, which is about proteins, what proteins are. So proteins are, sequences of amino acids. They each, unlike DNA sequence, each position on the, on the protein sequence can take one of 20 different values. Sorry, this is just some video. I thought that might help if. Okay, so this is a protein sequence here. Each position in the protein sequence can take one of 20 different values. So it can be one of 20 different letters of the alphabet. And this amino acid chain folds once it's formed once it's out of the nucleus, it folds and forms this complex 3D structure. And if you zoom into a particular part of this 3D structure, it's going to have a bunch of atoms, and mainly carbon, nitrogen, sulfur, and oxygen and hydrogen. These are the atoms that are present in all amino acids, and hence they form the main structure, the main atomic structure of every protein. So there are around 200 million known proteins, and every year around 30 million new proteins are found, are discovered. And as you are all probably already familiar with, the 3D structure of the protein is very important. It determines what functions that protein has, what it can do. And so it's important to know what the structure is. It's important to be able to guess what the structure will be given just the sequence. It's also interesting to solve the reverse problem, where you're given a structure and you want to know, not the exact structure, but you're given, say, some parts of it. For some protein engineering tasks, say you are interested in certain types of structures, and then you want to know what the sequence should be so that I can produce that structure. So all these problems are very interesting and important, both from drug discovery, from protein engineering and protein design perspective. So one of the most popular problems is protein structure prediction that I just described, which is solved by models like AlphaFold 2, from DeepMind, Rosetta Fold, and so on, where you want to know, given the one-dimensional protein sequence, what the 3D structure will look like. There's protein interaction prediction, where you want to know, given two proteins, are they going to bind to each other or not? Then there's computational protein design, where, which I just described again, you're given some desired structure. So you have some coordinates that you're given, and you want to know what amino acid sequence can produce those coordinates. So this is very interesting in protein engineering, in making enzymes for
table. We'll just continue speaking in the interest of time.
So,
That's fine.
okay,
In the
sorry.
interest of time, we can just continue.
So, yeah, so there are many different ways to represent. So the data that we get from this protein data bank, it has the coordinates of the atoms, the atom type, carbon, nitrogen, oxygen, sulfur, or hydrogen, and then which you can infer which atoms are neighbors of each other. So you can think that the various, as you can imagine, there are different representations. You can create a 3D grid, or you can think of it as a graph where the nodes are these positions, the atom positions, or they can be more coarse-grained positions, which is just the alpha carbon of the protein. That one is the node, and the edges are which others are the neighbors. So if a particular other atom is within, say, 5 angstroms, then in the graph neural network, you would connect those two nodes to form an edge. So here's an example of a 3D grid-based representation. So this is showing the neighborhood of this, the amino acid number 2 from this coronavirus protein. And this is the neighborhood. These are the oxygen atoms in that neighborhood, carbon, nitrogen, and sulfur. So you can think of them as different channels in okay, I didn't manage to join, sorry. Yeah, so you can think of them as different channels, the way you do it with image-based deep learning models, where you have RGB channels. You can have channels based on the atom type. And using this, you can train CNNs, 3D CNNs for any problem of your interest. So if you have a bunch of protein structures and you have a bunch of different types of labels, then you can train a supervised CNN-based model using a representation like this. So the particular problem that we try to solve here is that we want to find cryptic pockets, which are basically cavities that open up in a protein once it's in solution. So in the crystal state, you don't see the cavity, but then once it's in solution, once it's inside the body, because the protein is constantly moving, it has lots of different conformations. There exists some other conformation where the cavity is available, like this blue region here is the cavity that opens up, but it's not available in the original structure. So a lot of people use molecular dynamic simulations to study this, where you have available at time t=0, you have the crystal structure. And then once you run physics force field based simulations, you might see some cavity opening up. So this is the problem. Given the structure at t=0, we want to forecast which parts of the protein will open up. And this is an open problem in structural biology. So we train machine learning models for this problem. Just a quick overview. Why are cryptic sites important? Because they give us targets for drugs. Parts where there are pockets and cavities opening, they are nice candidates for drugs to go and bind there. So you can develop more drugs as a result of if you know what are the cryptic sites. So the way we set up this problem is we have this time window. We have molecular dynamic simulations data where our collaborators took this starting t=0 structure. They ran simulations for 40 nanoseconds. And then we observe what all has happened in those simulations. In some cases, some pockets open up. In some cases, nothing happens. And we use this molecular dynamic simulations data as training data. So what we do is we take this structure and we mark which parts are the ones that opened up. Those are our positive examples, and the parts that did not open up, they are the negative examples. So this is the supervised learning task, that given the structure at t equals 0, and the positions that opened up labeled as positive, positions that did not open up labeled as negative, we want to forecast this for a new protein, which we haven't seen in the training data. So this is, we use a graph. We develop a graph neural network, which is based on a prior work called geometric vector perceptron. The nodes are positions in the protein, edges are based on neighborhood, and what we want to predict is this. So if it can result in a cryptic pocket or if it won't result in a cryptic pocket. So it's a multi-label output. For each position on the protein, we produce an output. So the model that we use, the graph neural network based approach, geometric vector perceptron, it's equivariant and it's invariant. So it solves all these issues with rotations and stuff that I mentioned before. This is our training data. We had over 1 million different examples. And so it's very important to see how we split the train test. We want to make sure that proteins in the training data are not too similar to any protein in the test. Otherwise, it can overfit. And this is the prediction performance that we see. So we compare it with 3D CNNs. Here on the left, we have just standard ROC curve, area under the ROC curve. And on the right, we have area under the precision recall curve, which we do because there is some class imbalance between the positive and negative classes. And we find that the graph neural network has a somewhat better performance than 3D CNN. And on a completely held out set of proteins from a different source, we see that the graph neural network does better. Okay, so that sort of brings me to the end of this talk. And I want to thank and just end real quick with a little bit about the sort of things that my lab at Microsoft does. So there's a lot of work on bioacoustics, where we take audio recordings from whales, and we want to know what whale species it is. Some other people in the lab, they also develop models based on geospatial data, so satellite imaging data, trying to find solar farms from satellite imaging data. The general AI for health work consists of a lot of medical imaging. There's many different types of medical imaging work being done, PET images, fMRI, and so on, CT scan, and so on. And then there's protein structure-based methods. Okay, I want to thank you all. If you have any more questions, maybe I can take them now. Thank you.
I think we
Yeah.
what type of IT infrastructure takes to this type of
Yeah, that's a good question. So the first part of the work, we intentionally choose an approach that's not very compute intensive. That's one of the key selling points of that work. That it can, the variational autoencoders that I showed in the first part of the talk, they can be trained on a CPU as well. Like it takes around 2 to 3 days to train on in a CPU only environment, and it takes around 3 hours to train with one GPU, standard. I forget the configuration, but nothing very fancy. Whereas the second part of the talk is very compute intensive for the generation of the training data. So molecular dynamic simulations were generated from using GPUs, and that was like months and months of very large scale GPU compute for the molecular dynamic simulations. But the machine learning part, which was the graph neural network, that one takes 12 hours to train with 2 GPUs. So still very much within the reasonable realm. But yeah, so some other recent work that I'm doing, that one takes that's very compute intensive. That one is more of this very large scale structure, which was there in this slide deck, but I didn't plan to talk about it. Where we want to go from sequence to structure and structure to sequence very quickly. So we develop these diffusion models, which are kind of based on this framework called Dolly, if you have heard of it, from OpenAI, where you can give it like text and get the image represented by text. You can give all kinds of crazy text and vice versa. So we adapt the framework to work with protein sequences and structures. And that one is very compute intensive. We take around, we take 3 days to train with 8 GPUs. And even higher end version is this DeepMind, AlphaFold 2 type of model that we train for another problem. That one takes 64 GPUs and it takes 1 month to train the model. So there's like very different ends of the spectrum. It depends on how, so that is all transformers. So all of our transformer model based works, they take many GPUs and take weeks to train. But others are reasonable.
Someone put out on Grand Challenge, because I've seen very few multimodal problems come out on Grand Challenge.
Yeah, I agree.
They're very quite formidable. So all kind of problems applied.
Yeah, totally. Yeah, so we just adapt that to sequence and structures.
So these are open challenges.
Yeah.
Are you dealing with proprietary?
No, this is all open. This is all PDB, Protein Data Bank. This is actually part of a collaboration with this faculty called David Baker, who is like a big shot in proteomics. He's at University of Washington. So they have processed all the data from Protein Data Bank, filtered things, structures that are not good, but it's all publicly available, nothing proprietary.
Maybe another question. Why
Yeah.
is Microsoft doing it in the result?
So there are teams within, several teams within Microsoft that work on, in the health space. My team does very publicly facing work, sort of somewhat non-profit. So we collaborate with organizations where we have in common interests. But then there are some teams within Microsoft Research that are getting into proteomics. There have been several teams working on medical imaging technologies, on like pathology images and so on. Also on EHR data, which is like this electronic health records data in the US. So taking like a sequence of electronic health records from patients and trying to forecast. So that's like a more of bio NLP in some sense. So there are some teams at Microsoft Research that do those type of things. Yeah.
Do you think use different conformation of the same protein for training?
Yeah, that's a good question. So we have several trajectories from each protein. That's the way the molecular dynamic simulations work, because we don't know where we may see a pocket. So what we do is we start with the structure that we get from the database from, so PDB is the publicly available database that has like one structure for one protein, which is from the crystal. So we start the molecular dynamic, the force field based models with that structure. We make changes based on force field equations, which produces like a bunch of options. It produces like a trajectory. Some of those are sampled, and again, some more are run off as offshoots from there. So we have like a large number of these, like you start with one trajectory, then you have several routes going on from there, then you have some more going on from here, and so on. So yeah, for a single protein, we have several trajectories. In many cases, thousands of trajectories, and so training data will consist of from all of these trajectories, locations where we saw the pocket opening, and we see that, say this is the protein, and this is a particular location in the protein. Then we see how often was this appearing in pockets. Say it was generally not appearing in a pocket, maybe out of all this several nanoseconds, like hundreds of nanoseconds of data, only in 2 cases we saw the pocket very briefly, then it's going to be a negative. It's a positive if you see it in a certain fraction of the simulations.
So my question is, say for example, you use the protein catalase from E. coli in your training.
Mhm.
So would you test it only for catalase, or do you use catalase from different species for training? How do you?
That's a very good question. So we restrict to only human proteins and coronavirus proteins and some bacterial proteins, because that's what we have from our collaborators. So this is some people at University of Pennsylvania. This is like a biophysics lab that we worked with. So these are the 3 organisms where they have proteins, and we make sure that the proteins in the test data are very different from the ones in the training data. So like beta lactamase will never appear in the test data, even if it's from a different species, because then it's a somewhat trivial problem, because typically, because the proteins are conserved across species, at least some parts are very well conserved across species, you can expect similar pockets to open up in, even if it's in a different species. So we make sure that the sequence similarity is less than 30% in between our train and test. If it's greater than that, then it becomes like cheating. Go ahead.
This is from the PDB where you already have the structure. What happens for COVID, like there would be novel sequences where we do not already have a structure. What happens to that?
Yeah, that's a very good question. So what we do is we use a bunch of sequences and run them through AlphaFold 2. So AlphaFold 2 is the DeepMind model which predicts the structure. So we apply this model on like on thousands of sequences for which there's no structure on PDB, but AlphaFold 2 gives us a predicted structure, and then we run this, and then we test the model we have trained, and apply it on it.
How many pockets generally get where there is?
Yeah, that's a
Too many, right?
no, not too many.
Filter that you say that categorically this is more potential pocket than other, something like that. So that you cannot get a number of drugs.
Yeah, that's a great question. So it's a somewhat like, since it's an open-ended problem, the opinion is very diverse. Some people, like for instance, the lab that I work with and some of their other collaborators, they think that like 80% of the proteins out there have cryptic pockets, which is like an extremely, I think it's an overestimate. So 80% of all proteins should have some cryptic pockets. I don't think that's true. I think it's probably, and then there are others who think that maybe a few hundred proteins over the millions of proteins have cryptic pockets. So there's a lot of, there's no consensus yet, because most proteins are only known in their crystal form, which is on PDB. Molecular dynamic simulations are only available for a fraction of those. So there isn't a way to know, unless you do like a screen, like you said, like a very high throughput screen, drug screen, where you try like millions of different compounds on a single protein. Unless you do that, you don't have like gold standard information to know whether, like, how many cryptic pockets are there.
Suppose if the protein is 550 pockets,
Mhm.
so drug library screening, you see that 3 or 5 10 pockets are
are actually bound by something. Yeah.
Then you see that how those 10 pockets are different from
Yeah.
the rest of the 9.
Yeah.
Something like that.
Yeah, but I think the number is something like 3 per protein. We find something like 3 to 5 cryptic pockets, not more than that. No, even in very large proteins, you will see like, 3 or 5, 4 or 5, something like that. Fewer than 10. Okay. Can you ask the questions, please?
Yeah. Sure. Can I ask?
Yeah.
Yeah. So one question is, you spoke about sequence similarity between train and test, but if you look at structurally similar proteins, then the average sequence similarity between structurally similar proteins is roughly just 10% or so, right? So it's possible that you have structurally similar proteins in your train and test set. And so the question then is, does that make your problem easier in some way? That's the first question. The second is on the training set. It's known that a lot of cryptic pockets, at least as per my understanding, a lot of cryptic pockets open up only when there's a ligand close to the protein.
Yeah.
And it's that
Yeah.
protein ligand interaction
Yeah.
that determines the opening up. And so if you
Yeah.
do a free simulation of the protein in solution, you won't see them. And then
That's a
the
very
last
good question.
And then
That's
the last
actually,
question is really about the stability of the cryptic pocket, whether 40 nanosecond simulation is enough to
Yeah,
find
that's
stability.
yeah, sure. So the question, so maybe I'll start with the second question that it's Alpan, right? Alpan asked. So
Yeah.
it's that usually many cryptic pockets, they open when a suitable ligand, like a chemical compound or another protein is available. They don't open up if you do standalone molecular dynamic simulations, and which is the case. And this is something that we were also concerned about when we use this data from our collaborator. But then, doing molecular dynamic simulations of both the protein and chemical compound
It's tough.
together, it's tough because, first of all, there's just so many different options for the ligand, right? Like what ligand? Some groups do MD simulations on focused groups of ligands, like you pick a particular family of compounds and do simulations. But again, those are not exhaustive. So that's the catch with this. And what some prior work has done is they, rather than using any possible PDB structure, they limit the PDB structures that they use to ones that are known to be bound by ligands. And only those are used as the gold standard positives. So this is also something that came up in the reviews. So one of the reviewers asked the same question. The paper is currently under review in Nature Communications, and this is something they asked too. So it is a critique of this method, certainly. And going back to the first question, which was, we only filter based on sequence similarity, which is 30%. But then there might be cases where the structures are similar, but the sequences may not be similar. That is certainly possible, that there might be some domains, where, like say there are some alpha helices that are present in two different proteins.
I mean, if you choose a database like SCOP, for example, where you have structural families, and then split your train and test by structural families rather than sequence similarity.
Yeah, exactly. So we use two criteria. One is based on sequence similarity, and one is based on this database called SCOP. It's called Structural Classification of Proteins, SCOP. So the SCOP gives like a hierarchy of annotations. SCOP is like a hierarchy where each protein is assigned to like one part of the hierarchy based on what it does and its structure. So we make sure that the proteins are split also based on the SCOP topology. So topologically, they are not in the same class.
Okay, thanks.
Sorry, I didn't mention that because I wasn't sure if people are familiar with the topological classification.
This clarifies things. Yes. No. My last question was really about stability, but I don't think there's much you can do with what, given the limitations of MD simulations.
Yeah. So stability of the protein or of the
of the
model?
cryptic pocket, of the cryptic pocket. So
Yeah.
it could be that many of
Yeah.
the cryptic pockets that you discover, that you use as ground truth are actually unstable
Uh-huh.
because 40
Yeah,
nanoseconds
that's
is not
a
enough
question.
time.
So, yeah, so we did consider limiting based on duration. So, because we run the simulation, we know how many nanoseconds that pocket was open for, and how, we did consider using that as a way to establish like a confidence score, but that was limiting our training data a lot. Like a lot of data had to be thrown if we based it on duration.
Okay.
So getting a bit
Thanks.
too long.
Yeah. Yeah. Okay. Yeah.
Probably we should end here with applause.
Yeah.
Thanks.
Thanks. Thanks a lot for
Thanks.
the questions.